Hello students, this is Dr. Faizan Mirza. I am discussing the solved answers of October-November 2021 variant 11 of your MCQ paper of 9700 A-level biology. I'll be discussing question 1 till question 20 in this video, and the remaining half of the paper will be discussed in another video. So here, this is the paper. This is variant 11 of 9700. As I told you, I'll be discussing this paper with you, the first 10 questions. So let's start with the first question here. We know the first 10 questions normally when the MCQ papers, it actually comes to you. So the first chapters, they come in the first half of the paper, and the last chapters come in the last half of the paper. For example, the chapters that are from cell structure, microscopy, biological molecules, enzymes, cell membrane and transport, be it a mitotic cell cycle, nucleic acids and protein synthesis. Normally these questions come from question 1 till question number 2022. And yes, there are overlapping questions. There are questions in which this question will be of cell, but it will be taking into a consideration or into account some structures from plant transport or from gas exchange or from heart, for instance. But yes, the main premise of the question will stay cell structure or biological, whatever, as I've just mentioned, from chapter 1 to chapter 6. And then the next half of your MCQ paper, that's the remaining question. They are from the remaining part of your syllabus, which include the chapters or topics from transport in plants to transport in mammals, then infectious disease, gas exchange, and gas exchange. Now we do not have smoking, so yeah, gas exchange, and after that you have immunity. So all these, this is how your paper actually is divided, how the 40 questions are divided. So let's start with the first question. The first question here is very straightforward question regarding microscopy. And you can just, what I recommend to my students, and I'll just be highlighting this to you as well, that whenever you are solving MCQ, you should always annotate on the paper. You must have a pencil in your hand, which you will be having obviously to shade the appropriate answer which you are about to shade. But make sure that you do mention things on the question paper so you know what you are going and what you are actually working on. You must annotate. So let's move forward. A student observes cell using my light microscope. So let's recall what when you are using light microscope, you need IP reticule, you need a stage micrometer. I just mentioned this by recalling. Further, which items will the student need to calculate the magnification of the drawing? Yes, eyepiece reticle will be required, shade micrometer will be required, ruler will be required for measuring across the drawing, because when you draw something, you have to measure it across its width. So yeah, ruler is required, but hand length is of no importance. So yeah, our answer in this case will become 1, 2, and 4. So we can just move on from there, and we know that the answer is B in this case. So let's move to question 2 here. Now, question 2 in this case is actually asking you about which equations correctly show relationship between magnification, image size, and actual size in microscopy. We can recall the formula: magnification is equals to image upon actual. Just rearrange this formula. If you arrange the formula and image upon actual, you take actual there and magnification here. The formula becomes actual is equals to image upon magnification. So you just rearrange the formulas and see, and you can just see that the second option, the fourth option cannot be, the formula just cannot be rearranged in any way to get the second and the fourth option. Yes, first and third option are correct. So our answer will be 1 and 3. Let's move on to the next question. Which cell structures does DNA replication, DNA transcription occurs? So DNA transcription, let's recall DNA transcription. We know DNA transfer, you have template DNA. The template DNA turns into mRNA. Which structures have DNA? Nucleus having DNA. Yes, the DNA is linear. So I just mentioned this, okay, linear DNA. Why? Because I need to recall what nucleus is having. So chloroplast is having circular DNA. So I just mentioned circular DNA there. And mitochondria is having circular DNA as well. So yes, if there's a DNA and then the template DNA will be turning into an mRNA, transcription is definitely occurring. So the answer becomes D. Now, which row could correctly identify nucleic acids protein, 2 different virus particles in sample of air? Now, very common error that students do in a question that comes from virus, and something that students have a confusion with, or a very common thing that they ask or they check. Students should make sure that they remember virus cannot have DNA and RNA together in its structure. So it's either the DNA or the RNA. Both of these nucleic acids can never be present together inside a virus, in a viral particle. So here you are having the viral particle, and the viral particle is having DNA, RNA. Both know that's an incorrect option. If both are absent, again that's an incorrect option. Second and third options are correct. That yes, if DNA is present, then RNA must be absent. If DNA is absent, then RNA must be present. So in this case, the answer becomes C, which is 2 and 3. Biological molecule question comes next. Is the you took 4 samples of biological mixture molecules and they gave the following results. The tests are shown to you. You boiled with excess Benedict solutions and the color stayed blue. That means reducing sugar is absent. You just annotate. You just mention here with pencil that okay, reducing sugar is absent. Now you carry out acid hydrolysis, neutralization, and again checking with Benedict solution. Now the color turns red. It means that the non-reducing sugar is present. So yeah, that's present. Biotech reagent, you test a protein, purple, the color changes, so protein is present. And ideal solution, starch is absent because the color stays yellow. So I annotated all the readings here. And when I took out all these readings, so I know that by reducing sugar is absent. So every option which is having a reducing sugar in the answer, so that's not the option. And since starch is absent as well, so that's not the option either. So my answer is C. One molecule of X is formed by a single condensation reaction, releasing one water molecule. What is molecule X? So if only one water molecule is being removed, single condensation. If one water molecule is being removed, then definitely it must be a disaccharide because we know when two monosaccharides combine together, a water molecule is lost. A water molecule is lost. For example, if you are making a triglyceride, 3 water molecules are lost there. Question 7. The table shows for information about carbohydrate polymers, which will describe glycogen. Just recall what glycogen is. I quickly just mentioned its branch. It's having alpha 1 4, it's having alpha 1 6. It store energy or store glucose, not energy. It's a long-term storage molecule, so it stores those glucose units. So if I just with this annotation, I check, I quickly come to my answer that okay, this is the answer. Why? Because it's a function of the molecule. It cannot be structural. So C and D cannot be my answer. It is not helical either. So my B option is cancelled as well. The only answer that's viable for this option will be A. The diagram show for 4 phospholipid molecules. Which molecule could contribute most to the fluidity? You just recall what most fluidity actually looks like. Most fluidity would be with carbon-carbon double bond, short fatty acid tails, cholesterol, high temperature. Just recall what actually increases the fluidity of the membrane. Now, the options are not taking into consideration any of these. The question is taking into consideration only the carbon-carbon double bond. So wherever you see this double bond, this is a double bond, this is a double bond, these are the double bonds. So here you have no double bond. So this cannot be the answer. Here you have one carbon-carbon double bond. Yes, this might be an option. This is having 2 carbon-carbon double bonds. So obviously this can't be the answer. This can't be the answer. This might be the answer. But option D is having 4 carbon-carbon double bond, the most carbon-carbon double bond. So the more the number of carbon double bonds are there, more fluid the membrane will be. So most fluidity will be because of this, and least fluidity will be because of this. They may just reframe the question next time and they can ask you which of these can change the fluidity to be the least. So yeah, in that case, A would be the answer. But in this case of most fluidity, the answer is D. Which property of phospholipid molecules explained by a single layer of phospholipids added to water immediately form bilayers? This question is simply factual question. You can just recall that hydrophobic fatty acid tail, fatty acid chains, they repel water. Yes, they do repel water. The tails pack together. They do the molecules, they form micelles. You have studied that. The non-polar fatty acid chains are attracted to each other by hydrophobic interactions. Yes, because non-polar fatty acid tails do show hydrophobic interactions. Hydrogen bond formed between phosphate group and water. Yes, because both are charged entities. They do interact. And yes, the answer will be A, 1, 2, 3, 4. 1, 2, 3. No, there is no 4. Okay. A, 1, 2, 3. Question 10. The diagram shows 3 examples of different bonds. So you can see 3 bonds here. What I did when I was doing this question, as I'm very used to doing this, is I always annotate. I always keep a pencil in my hand. Now, since this was soft copy, I use my the pencil that I was having, and I just mentioned what these were. So this is bond 1. The bond 1, you can see it's a dotted bond. It must be a hydrogen bond because it's present between a hydrogen and an oxygen. Oxygen belongs to another structure and hydrogen belongs to another structure. So the bond between them must be a hydrogen bond. Now, carbon-nitrogen bond, you know this bond is a peptide bond because there are 4 atoms. Just recall there are 4 atoms which actually are attaching carbon and nitrogen together in a peptide bond. So this is a bond which is having 4 atoms. So although this is not asked in this question, but this is a commonly asked question as well. Third is a disulfide bond. How do you identify the disulfide bond? It's always present between sulfurs. And you can just recall that which amino acid is having sulfur, cysteine residues, cysteine, CYS, cysteine residues. If 2 cysteine residues, they are present one another in front of one another, the sulfur bridges together to form disulfide bridge. Now, which bond hold tertiary structure of proteins together? We know that the tertiary structure of proteins is all because of R group interactions. It's all because of side chains. You can just recall the alternate term, R group interaction or side chain interaction. And side chain interaction are because of hydrogen bonding and disulfide bond. It is never because of peptide bond. So in this case, the answer will not have option two. And you can see they, just to confuse you, they gave two in three of these options. So they confuse the students in such way in many a times, not always so, that the wrong answer will be given in multiple options to just confuse the students. So the students think, okay, two must be there in the answer. That's why they have given three options, because in O level we do have such MCQs in which an answer, if it's repeating, then the chances of that being the answer increases. But here, this is completely a trick of the examiners, so beware, do not fall prey to their tricks. Question 11, which of the following feature of collagen enables it to fulfill structural role in skin and tendons? Just we can cancel out the wrong options to arrive at the right answer. Adjacent collagen molecules are linked by ester bonds. No ester bonds are not present there because ester bonds are present in fat. Collagen fibers form layers with fibers in different directions. No, this is not in different directions. Collagen molecules are formed in triple helix polypeptide chain. Yes, that's an option, and that's actually the correct option. So yeah, that will be our answer. Polypeptide chains of collagen are tightly folded. No, they are not tightly folded. So try to always find the mistake in the stems of the options, whichever the mistake will be, because in that case, the chances of you arriving at the right answer will be very high. Now, this question 12 is a very interesting question and kind of a difficult question in which you probably would be spending more time than you should. But anyhow, keep in mind that this is still a 1 mark question. There are no distinction questions here. So spend the time that actually you smartly can. So question 12 is about enzymes. If the question is about enzymes, make sure that you do recall the graphs. And this is something very important because in enzymes, they give graphs as always to just see how good the student can analyze the graph. The graph will always have a y-axis and an x-axis. The x-axis will be having and the y-axis will be having some variables. You must know how the variable along the x-axis as it is being changed, how the variable along the y-axis changes because the x-axis being changed. For example, in this case, this is time given, this is concentration given. This shows that if you are increasing the time in this direction, the concentration is increasing. Now, there are 4 curves here. The question states the graph shows how concentration of 4 components, 1, 2, 3, 4, of an enzyme catalyzed reaction change with time. How much time is mentioned? It's not given. How much enzyme was added? It's not mentioned. How much substrate was added? It's not mentioned. How much product was from? It was not mentioned. This is a completely open-ended graph. The graph is not actually telling you that the completion of reaction occurred, because you can see that if we just come to this graph, we see that okay, there is something whose concentration was maximum at the initial time, then it started to decrease, then this must be the substrate, because we know as the enzyme catalyst reaction proceed along time along the x-axis, the concentration that falls will be always of substrate, and the concentration that rise will always be of product. So we know 1 is product and 3 is substrate. Substrate, can you see that did the substrate come to the x-axis altogether? No, the reaction is still going on. They ended this experiment investigation or the graphs here. The reaction is still going on. The substrate is not fully consumed. There is still some amount of concentration of substrate left when the reaction was stopped. Now we know that our answer cannot be 1, our answer cannot be 3. So A is incorrect, C is incorrect. Now we are left with B and D, which are 2 and 4. Now, what's 2? If you look closely at 2, what do we see? We see that initially the concentration is very high, in fact high, and as the time proceeds, the concentration decreases. Now, once the concentration decreases, okay, you may think that, okay, this might be enzyme substrate complex, because this is exactly what they are asking, which component is the enzyme substrate complex. So yes, you may think that, okay, 2 might be enzyme substrate complex because its concentration was high initially, and as the time proceeded, its concentration decreased. But how can you explain how enzyme concentration with substrate started to increase once again? You cannot explain this part of the graph using enzyme substrate complex concentration. Yes, this part may be correct, but the remaining part of this graph is very clearly not the enzyme substrate concentration. So what is this 2? This 2 curve is actually enzymes with the free active sites. What do you mean by enzyme free active site? When the reaction starts, you know that substrate is very high in concentration. This is substrate being high in concentration, product being 0. So all the enzyme active sites are free. This enzyme active site, they are free. As the time proceeds, the enzyme active site get occupied. So there are very few enzymes available with free active site. As the time proceeds, more product is formed, more and more enzymes, they free up their active sites again. So its concentration start to increase. So we have cancelled 1, 2, and 3. The only option that we are left with is 4, and 4 exactly is our answer. This is the line that you see that stays constant throughout. Now, when a concentration time graph is given for any enzyme catalyzed reaction, in most cases, in fact, in almost most all cases, the concentration, if it's very low, then this is a hint that okay, this just might be the enzyme, because a very low concentration of enzyme is required, and the concentration stays constant. If the concentration stays constant, then definitely this is the enzyme, because from the start of the reaction till the end of the reaction, the concentration stays same. There's the enzyme. So what about enzyme substrate complex? So in this case, since 1, 2, and 3 are not our options, this line represents enzyme substrate complex as well, because we are not told in the question what exactly was the substrate concentration. Was the substrate in excess? We assume yes, this was in excess. We know that the enzyme is in limited concentration. In that condition, we know that the enzyme quickly becomes saturated, and it stays saturated throughout the reaction as the substrate keeps on decreasing. Now, if the graph was shown to you to decrease the substrate concentration to x-axis, then probably the curve for enzyme substrate complex might have shown this to be decreasing as well. But as I told you earlier as well, this point tells you that this investigation was ended at that point. So the substrate is not fully consumed, the products might still be forming, and the experiment was ended. This is a lengthy working in this question. Keep in mind that this question is still of 1 mark, so you should not spend more than 1.5 minutes on such questions. If such questions come, if you have such a question where you are spending a lot of time, then just move on to the next question, because there will be a lot of questions ahead in this paper, which you can do in 1.5 minutes, and then come back to this question later. So let's come back. Question 13. So let's move on. Now in question 13, you can see the table shows Michaelis-Menten constant. What do we know about Michaelis-Menten constant? Just recall Km, whenever Km is high, that means the affinity is low. So if you look at the exponent values here, we know that this enzyme C is having the highest Km value, and F is having the least Km value, because this value is given in exponent of 10 to the minus 6. You just shift the decimal 6 places back. So if this detail is kept in mind, enzyme F has the greatest affinity for its substrate. This is exactly what we are looking for. This is exactly what we are looking for, because the greatest affinity of the substrate is the enzyme with the affinity. We know that Km affinity, how they are linked together. If the Km is high, the affinity is low. If the affinity is high, the Km is low. So enzyme F is having the least affinity. So sorry, the least Km value. That means the affinity of our substrate is greatest. And the reverse can be said for enzyme C. Enzyme C is having the highest Km value. So in this case, it will have the least affinity. The question might be reframed in another paper or somewhere ahead. They may just ask you about this. So there's no issue of getting confused there, because an enzyme whose Km is given to you as being highest, it will always have a low affinity for the substrate. Option A is actually telling you about Vmax. Option B is about Vmax as well. Option D is about Vmax as well. So what you can just remember and what you can just recall, that there is no link of Km with Vmax. You can't predict Vmax with Km. So option A, B, and D will be outright incorrect, because they actually take into consideration Vmax. Okay, so coming to question 14, what explains how signaling molecule produced by one cell can be detected by a target cell? So we know that when a cell signaling molecule is producing a cell signaling molecule being produced, the molecule will always have a shape complementary to its receptor. So it can bind to any type of cell. No, it cannot bind to any type. There are no universal signaling molecules as such. Signaling molecules will be having a complementary shape to the cell receptor. Yes, perfectly. Signaling molecules enter cell by diffusion. No, only the non-polar signaling molecules can enter by diffusion, since there is nothing mentioned where there's a polar molecule or a non-polar signaling molecule. So this option C is incorrect as well. Signal molecules enter the cell by endocytosis, so polar molecules never enter. So you can just very well with confidence say that the answer of that question is B. Now moving on to question 15. Some enzymes are produced by cell in pancreas. The enzymes are secreted when required. So if it's secretion, the question answer is actually all about exocytosis. So yeah, that's exocytosis. Plant cells were submerged in a solution with a water potential that have less negative than found inside the cells. What do you mean by this? Less negative than that found inside the cell. Less negative means there is more negative water potential inside the cell. So when there's more water potential inside the cell, more negative water potential inside the cell, water enters the cell. So when water enters the cell, can the cell plant cell burst? No, the plant cell cannot burst because it's setting the cell wall. Would plasmolysis occur? No, plasmolysis occurs when the water leaves the cell. So the only option left is turgid, and that's basic just the fact of osmosis. What would be present in each chromosome at the end of G2 phase of the cell cycle? So two centrioles, no, there are no two centrioles present. There are no two centromeres. Two molecules of DNA, yes, DNA replicated in S phase. And if the DNA is replicated, we know one linear DNA molecule is having two telomeres, and this is the telomere, and this is the telomere. One linear DNA molecule is having two telomeres. So if the DNA underwent replication in S phase, then there will be two DNA molecules. So the two DNA molecules will have four telomeres. So the option D is incorrect as well. This will not have two telomeres. Yes, there will be two DNA molecules because the DNA replicated in S phase. Question 18, which events listed are a part of cell cycle? All of these are a part of cell cycle: interphase, metaphase, cytokinases, because metaphase is a part of M phase. So yeah, that's a part of cell cycle as well. Question 19. Now, question 19 again takes into consideration a common error that the students end up doing, and which features of the organism are affected by drug that kills the cell that are dividing mitotically. You need to follow the idea that the cell is getting killed. So because of the drug. So which of the features will be affected? Will cell repair be affected? No, cell repair will not be affected. Cell replacement, number of stem cell, tissue repair, tumor formation will all be affected. Why? Cell repair never occurs by mitosis. Keep that in mind. Cell repair never occurs by mitosis. It occurs by getting rid of old and worn out organelles and of producing new organelles. That's what cell repair is. There is a huge difference between cell repair and tissue repair. Cell repair and tissue repair, they are two different things. Cell repair means the cell getting repaired from inside, and tissue repair means the entire tissue is getting repaired. Yes, mitosis will be required there. Now, the last question for this video, question 20. The question 20 here takes into account the question says, graph show the distance between sister chromatids curve P and the distance between chromatids and centrioles curve Q during changes during part of mitosis. Now, when such information is given to you, always just annotate on the diagram what is line P. So line P actually is telling us the distance between sister chromatids. If the distance between sister chromatids, if the distance between sister chromatids, sorry, this one, if the distance between sister chromatids, this line here represents distance between sister chromatids. This is distance between sister chromatids. What's happening here? The distance between sister chromatids from the time W is increasing. Y-axis shows to you distance in micrometer, x-axis shows to you time. So as the time is proceeding, the time at time W, this distance between the sister chromatid, it started to increase. Now, when would it increase? When would it increase? You can just recall that this point must be metaphase, because at metaphase, the sister chromatids, they start to separate, and in anaphase they start to move. So this is where anaphase is proceeding, and with anaphase being proceeded, the sister chromatids are pulled apart and the distance between the sister chromatids keep increasing. Now, coming to this line actually tells you this is curve Q. They say this is the distance between chromatid and the centrioles. So distance between chromatid and centriole is constant from time T till time W. The distance between the chromatids and centrioles stays constant, and at time W, the distance between the chromatids and the centrioles start to decrease, because this W is metaphase. Centrioles, they start to pull chromatid towards themselves. When the centrioles, they start to pull them chromatids towards themselves, the distance between them start to decrease. Metaphase and anaphase, just recall, when metaphase is actually proceeding to anaphase, the sister chromatids, they are pulled apart. Just basic recalling of facts here. Which statement could be correct? T and T to V represents interphase. If T to V is interphase, this is just not possible because chromatids do not exist in interphase. The DNA exists in the form of chromatin. So this is an outright wrong statement. T into W represents metaphase. Yes, this option is correct because throughout metaphase, the distance between chromatids and the centrioles, they stay constant because the chromosomes are lined up along the equator of the cell and they are all lying there held by the spindles. W2X represents anaphase. Yes, W2X represents. And so anaphase already, as you have seen, yes, 2 and 3 are correct. And the answer will be B. That's it from my side in this video. Catch up with me for the next half of the paper. That's it from my side. Thank you so much.